welcome to the next episode of my science podcast. Today I'm delighted to welcome Dr. Oliver Pearce, who is a biochemist whose research focuses on the tumour microenvironment, understanding the composition and function of the tumour extracellular matrix in immunosuppression, and the tumour matrisome's role in tumour immunity. And in September 2017, he started his own research programme with funding from UCB Pharma and against breast cancer. So thank you so much for joining me today.
Nice to be with you, Martha. Thank you for having me.
So I guess to begin with, may you give us an overview of your career so far?
Yeah, sure. So I always had quite a strong interest in science. From quite a young age. I had a chemistry kit when I was 10 or 11 years old, and I actually started out my more sort of research career in chemistry. So I was very interested in organic chemistry when I was younger, and I had the opportunity when I was doing my degree to go and work for a pharmaceutical company called Merck Sharp and Dohme, and basically what that did was expose me to a very high level of research. So suddenly, from going from sort of university level equipment, I was suddenly given industry level equipment to work with, and it turned out to go really, really well. And one of my tutors at my university suggested to me that I should consider doing a PhD, which I hadn't really considered up to that point, honestly. But after I finished my degree, I actually went back and worked for the same company for another year, and they said to me again when I was working there, look, you should go away, get yourself a PhD, and then come back and work for us at a slightly higher level. So I went away and did the PhD, and I never went back to industry, just because the research was so interesting. So the type of research I was doing at the time was focused around cancer. In fact, when I was working in industry, I was making small molecule drugs to treat cancers. So I kind of kept that whole cancer theme throughout my research career. When I did my PhD, I was looking at modifying the surface of viruses and using viruses as a way of killing tumours. And one of the molecules I was working with there was something called glycans, or often referred to as sugars, which is one of the major building blocks of life. And I studied that a bit more for 5 years as a postdoc, which is something you do after you finish your PhD, which I did in California for 5 years, and then came back to the UK and worked more in a cancer institute after that. And as you said, then started my lab. So my lab is all focused around targeting and treating breast cancers and ovarian cancers, but we take a slightly interesting angle in the sense that we don't deliberately target the malignant cells directly, but we're more interested in the microenvironment around those malignant cells. So we tend to look at other cells that are infiltrating into the tumour, and also we concentrate on something, or what we call the acellular part of a tissue, which is the proteins that make up a tissue. This is the matrisome that you mentioned earlier, and you can kind of feel the matrisome if you touch your hand, and you feel that elasticity in the skin. That's collagens and elastin in your skin. So these are the proteins which make up all of our tissues, and tumours express a particular type of tissue matrisome.
Wonderful. So before we go into the details for our viewers at home, may you define some of the terms which you've already touched on? But
yeah, sure.
So the ECM, something like that, the TMC, sialic acids, and Siglex, which we're going to talk about through the podcast.
Yeah, so the ECM stands for extracellular matrix, and so it's everything outside of a cell. And so there's some, you could sort of ask, well, what's more important, the cells or the extracellular matrix? And of course you need both. But you do obviously come across tissues where there's a lot of cells and not so much ECM, but then you'll find other areas within a tissue where there's lots of ECM and very few cells. So it's quite heterogeneous. Tumour microenvironment, I think, is the other one you asked for, which is the TME. So this describes just the local area within a tissue where the tumour is growing. So something that people might not be aware of is when we talk about tumours, people probably think of just a rogue cell, a cell that's dividing uncontrollably, which is what a tumour cell is. However, actually, if you look at a tumour tissue, only about as little as 30% in some cases is actually made up of these tumour cells. The rest is all of this other immune cell infiltrate, which is trying to regulate and kill the tumour. There's other cells there like fibroblasts, which are trying to respond to the tumour as it's growing in the tissue. So a lot of the tumour is in fact not malignant cells. In some cases, it can be lots of other things. Sialic acids, so these are a type of glycan. So they're one of the building blocks of life, and the expression of these types of molecules changes quite a lot in disease, and it has quite a big impact in how our host bodies recognize tumours. So they're very involved in the immune response against a tumour. And Siglecs are part of a family of receptors. So receptors are found on cells, and they're a way for cells to sense the environment that they're in. Siglecs, in this case, are important because they specifically recognize sialic acid, patterns of sialic acid expressed within a tumour. Does that help?
That's perfect. So we'll get down to business. So may you explain what matrisome proteomics and glycomics are, and how can you use these to identify potential molecules that might play an active role in disease progression?
Yeah, so these are two terms for types of analysis that we do specifically in my lab, but there are other labs as well which are using these techniques. We've refined them a little bit. So the matrisome part means studying the extracellular matrix, and the glycomics part means studying glycans. And the omics bit of that means that you're going to analyze a lot of things in one go. So historically in science, you might have one molecule that you analyze, like perhaps there's a particular protein that a cell expresses, and you'd spend all your time working on this one protein. So you can imagine research is pretty slow because you're just working on this one thing. So what we've got now is all of these techniques which are based off of sequencing or using something like a mass spec machine, and you can basically analyze not just one thing, but thousands of things in one go. So doing proteomics and glycomics allows us to analyze all of the proteins in a piece of tumor tissue in one go. So it's quite a very powerful technique. And so we've developed this specifically to look at ECM proteins, because we think these are really important and playing an active role in disease progression. And we also use it for looking at the glycans. And the reason we're so interested in glycans is because these are molecules that decorate most of the proteins that are present within the extracellular space. You can imagine them like if you think of a brush on the surface, they kind of form this brush-like structure on the surface of proteins. And if you think about how a cell interacts with its surroundings, cells also have this big brush-like structure of glycans on their surface. When the cells interact with each other and with proteins within their extracellular space, they touch first through the glycans on the surface. This whole thing together is called the glycocalyx, another term just to confuse everything a bit more. So importantly, they're probably heavily involved in the immune response, particularly in a tumour. So that's one of the reasons why we have quite an interest in glycans.
Okay, so how does the tumour microenvironment stimulate extracellular matrix remodeling, expansion of abnormal vascular and lymphatic networks, and migration of cells into and out of the tumour mass?
Just a small question that you've asked there, isn't it? So I think it's a very good question, and I think there's a lot of ways you can think about it. So in terms of how tumour stimulates this production of matrix, it's interesting because some people may argue that this matrix starts to be produced before a tumour's even arrived. So something that we would call a metastatic niche, which simply means the tissue that a tumour can arrive at from the site from where it was first growing. And it's important to note as well, I think, that so it's metastasis actually is usually the problem in cancer, in terms of it's the thing that often leads to mortality. So particularly for something like breast cancer, where you could use surgery to remove the primary disease quite effectively, it's the metastasis that's usually a problem, because it ends up going to organs or tissues which you can't get to very easily by surgery, or their essential organs, for example. So, but to keep it simple, the matrix is probably generated by factors that are released by tumours, and that stimulates another cell type called fibroblasts. And the job of a fibroblast is to maintain tissues. So in response to these factors that tumours release, they start to remodel that tissue. And it's partly in response to, if you think about cells, and in a normal situation, in a healthy person, cells have to know, very basic things like which way's up and which way's down. And a malignant cell loses that ability. It doesn't know which way's up and which way's down, and that's why you start to get this weird, invasion of cells into a tissue. And that matrix response, I think, is in part to that invasion, because it's damaging the tissue. It's like a wound, and so matrix is laid down in fibers to try and heal that wound. And it's fibroblasts trying to do their normal job of maintaining tissues. So, for example, when you cut yourself and you need to heal that wound, that will involve cells, fibroblast cells. They will do that healing process. They're trying to do it in a tumor, but it's just not having the same. It's just not having the same outcome, unfortunately. And then you've got other layers on top of this. So I think you mentioned the vasculature. So you need that vasculature. So tumor needs a vasculature to grow. There was some really, this is going back a long way now, some really old studies where they looked at post-mortem at people. And it turned out that, in fact, when you get to later in age, most people have some benign tumors growing in them, right? But they don't have a very good vasculature. So they can never get any bigger than, a few millimeters in size. So they're just not a problem. So the vasculature is essential for a tumor to expand and grow. Now, there's kind of like two trains of thought as to how you can interfere with that. So one idea is that you can try and block vascularization, but in practice, it doesn't work that well. I think tumors have too many counter mechanisms to help stimulate vascularization. On the other hand, one of the problems with the vascularization in a tumor is that it's very wonky. It's not very well formed, and so cells, particularly immune cells, find it very difficult to travel through the vasculature of a tumor. So one idea is that actually we should stimulate vascularization in a tumor so that you have quite a normal vascularization, and that would actually support immune cell infiltration into a tumor, and it would probably have a better host immune response and killing of that tumor. But so it's kind of, that's kind of cutting edge stuff that's going on right now. Some of the researchers in the center I work in focus on that side of things. And then finally, you've got lymphatics as well, just to add on top of the other levels of complex. So tumor is very complicated, but it really, all it's doing is it's all of the same processes that we have in a normal body. It's just that it's just slightly different in the situation of a tumor, because the way it kind of coerces and corrupts those normal processes. So the lymphatics has several functions in a normal healthy situation. Partly it's to absorb excess fluid in tissues, and then make sure that fluid then makes its way back into our blood system, so that you keep the blood levels correct. And it's also it's also sampling what's going on in our tissues, and it's really set up for regulating infectious diseases that might be present. So as these fluids are drained, they end up in things called like lymph nodes that you've probably heard of, and these are places where there's lots of immune cells ready, and there's another specialist cell called a dendritic cell, which is in your tissue going around sampling everything that's there. And occasionally it picks up something, that's foreign, a protein from perhaps, a bacteria, for example, or a virus, and it will display that on its surface, and it'll travel off to the lymph node via the lymphatics, where it will show this protein to another type of immune cell called a T cell, which will recognize it and start to proliferate to generate an army of T cells whose whole design is to just go out and kill this infectious disease. So that kind of thing happens in cancers as well. Happens in cancers, but tumors have worked out ways of overcoming that immune pressure. Does that answer your questions?
Absolutely, that's fascinating.
Yeah, good.
So my next question is, when profiling an evolving human metastatic microenvironment for high-grade serous ovarian cancer, as you've mentioned, how did you measure, well, lots of things all at once really, gene expression, matrisome proteomics, cytokine and chemokine levels, cellularity, extracellular matrix organization, and biochemical properties all on the same sample?
Very, very carefully, Martha. We did it very carefully. Yeah. So it was kind of a crazy idea, really, looking back on it now. But there's lots of studies which have looked at one of these things. So, for example, they've done sequencing and looked at the gene profiles. That's important because it tells you what's happening within cells. So you look at all these different gene profiles. It gives you an idea of what's happening within cells and then within a tissue. But no one had ever, at that time, no one had really tried to look at lots of different biological processes all going on at once and integrate it together. And we were interested in doing that. Actually, the reason we were really interested in doing that was because we wanted to put together a reference profile to help us build models, to build cancer models. So we needed a reference profile to say, what should this model look like? And so we did it all on human tissues. But the more we started to gather the data, the more we realized that there's a lot of consistencies here. So just to give you a short overview of what we did, we analyzed, I think it was about 6 different parameters on each tissue, and we split these into 2 groups. So we had things which we called like the building blocks or the molecular components, and these were the things like the genes and the proteins. And then we had what we called higher order features. So these are things that describe a tumor. So it could be, for example, its architecture. It could be how stiff it is, so like the physical properties of the tissue, and it could be the cellularity. And then what we did was we took all these things together and we integrated them together using a very simplified type of machine learning. So basically we had an algorithm running on a computer that integrated all these data sets together. And what we basically told it to do was tell us what are the key features of a tumor. And from that, it pulled out a very short list of matrisome proteins. So again, this is extracellular matrix that seemed to be very consistent across all of the tumor samples that we'd looked at. And interestingly, what we did with that was, so this turned out to be it's very small, only 22 molecules, which, if you consider you've got something like in the region of 20,000 protein coding genes, and they can have lots of different variants. So you're looking at, I don't know, 60,000 protein products that the cell can produce, and then you've got other modifications on top of that. To pull out just 22 things is quite remarkable in a way. And what we found was that these proteins were predictive of very poor outcome, not just in ovarian cancer, but in pretty much all of the other solid cancers that we looked at. So they're consistently expressed across lots of different cancers, which was kind of the exciting thing. And they're predictive of poor outcome, and they're also predictive of immunosuppressive environments. So this means they're associating with an immune system that is unable to kill the tumor. So we thought that was a very surprising observation that actually tumours, for all of their complexity and heterogeneity and differences from patient to patient and cancer type, that it all came down to these 22 molecules, I think it was, which are very consistently found across all of these different tumours.
Yeah, and that's going to have huge implications for, well, for cancer treatments in the future, because if you can then you know, target those 22, then that's something that you can aim for all of them.
Well, that would be lovely. I mean, if it turns out that, you know, one of the things with all of this type of analysis is that, of course, it's correlation and it's not causation, right? So you have to, in a sense, that's just what we did there was just a first step.
Yeah.
And what we have to do now is work out whether or not, you know, these molecules actually are having a direct effect in tumour progression. But I totally agree with you. If it turns out that, you know, let's say one of these molecules is really heavily involved in tumour progression, and we target it, and it turns out we can, you know, inhibit tumour growth. The lovely thing about it is that it could be applicable to lots of different cancers, which would be fantastic.
So you touched on this really quickly in your last answer, but in what way is tissue stiffness significant in these studies?
That's a great question. You know, I don't know if I have a really clear answer for you on this one. It's such a, it feels very chicken and egg to me at the moment with this. So when a tumour forms, as I kind of spoke about already, you have these fibroblasts try to heal the wound that's forming as a result of the tumour. And that leads to lots of fibrous proteins being produced, which are all kind of cross-linked. And that leads to a stiff environment. So it might just be that you end up with this stiff environment. On the other hand, these stiff environments have an effect on phenotypes. So for example, certain immune cells will respond to this type of stiff tissue and will change their phenotype as a result, which means, and by phenotype, I mean like how they're functioning. So they could end up supporting a tumor rather than inhibiting it, because they sense this stiff tissue. So I mean, we thought it was important because it's such a classic hallmark that you see as a tumor progresses, that they always tend to come out as being stiff. But I think there's another question there which I don't feel I have a good answer for, and maybe someone else does. But you know, how important is it actually to tumor progression? For example, if you let a tumor grow, but you blocked that effect of stiffness, would you really stop the progression of the tumor? And I can't think of a study that's actually demonstrated that, although I think that's probably the thinking in the field at the moment. So I don't know, but it certainly has an effect on cell phenotype. It definitely has an effect on immune cell phenotype. So if it turns out that an approach might be, for example, we need to change this immune cell into this type of immune cell, which is going to be inhibiting tumor growth, and we can do it by targeting the stiffness, which is possible, then that might be a nice approach. So it's potentially there's a lot of legs potentially in targeting in stiffness. I just feel right now I think we're just at the early stages of understanding what it means.
Okay. So my next question is, what is the matrix index, and what implications does this have for the future?
So yeah, so the matrix index is the name that we coined for this list of 22 proteins that we identified from the analysis. And the reason we call it an index is because it's quantifiable. So you can assuming you have a data set on a set of tumors, we can apply our matrix index to it, and we can say these people are going to be at high risk, these people are going to be at low risk. So it's quantifiable, and you can use it to make predictions about the data set. In terms of like what can you use it for? Is it useful? It's three levels. So I suppose firstly it gives you an idea of the tissue composition of tumours, which, up until then we had some ideas, but we hadn't done this kind of omics approach to give this broad overview. Secondly, it's in its simplest form, it's a list of targets that you can use to kind of explore further and say, when you've got a list, the matrisome proteins make up about something like 1,400, 1,500 members. So getting the list down to 20 makes it really manageable, and you can start to just do some exploratory work looking at these individual molecules. I think thirdly, something that I'd love to do, and we've not been able to get started yet, would be to consider using these as biomarkers. So one thing I haven't said is that these types of proteins actually end up circulating in blood as well, because they get degraded by enzymes that are present within the environment, and fragments of them end up circulating in blood. So it turns out you can detect them in blood quite simply. And we were thinking that perhaps these would be a good biomarkers to, first of all, predict the likelihood of perhaps a cancer being present, but also you may be able to use the pattern of expression of these proteins to say where the cancer is as well, which would be much more powerful. Definitely. So we'd love to develop it as a tool like that, but we haven't quite started that yet. That's something for the future, perhaps.
Wonderful. So this next question, I'm sorry if I mispronounce a few words.
Oh, don't worry.
So sialic acids can engage the myelomonocytic lineage inhibitory receptor Siglec-9, evading immunosurveillance. May you explain this in terms of upregulation of sialoglycoconjugates by tumours and the implications this has?
That's all good. I think you pronounced it all great. That's fine. Yeah. So these are so the idea that there's a change in sialic acids or the broader family of things called glycans, which I mentioned before is one of the essential building blocks of life. This has been known for over 100 years. It's like one of the first hallmarks of cancer, one of the first things that was observed back in like 1904 or something like that. It was one of the very first molecular changes. Of course, cancers have been detected. First recorded detections of cancers obviously go way back into ancient Greece. So they've been on the radar for a long time, but they've been very difficult to analyze and therefore also to work with as well. They're quite difficult to isolate and explore in the lab. Actually, I should sort of say, some of the pioneers who've looked into cancer glycans work here in London. For example, people like Joy Birchall at King's College London, for example, who's I think recently retired. Was a real pioneer in this field, identifying some of the early observations of changes in glycosylation in cancer. And also I ended up working with, when I was in the US in California, working with a physician scientist called Ajit Varki, who again is a pioneer in the field, and actually he coined the term along with some other colleagues for Siglecs, and has spent his whole life exploring, or most of his academic life exploring sialic acids and Siglecs. So there's been a lot of work in this area over the past, I suppose, what is it now, 50 years or so. What I was working on at the time when I was working with Ajit was looking at how Siglecs and sialic acids, so Siglecs expressed on macrophages, were interacting with sialic acids produced by tumours. And we've discovered from that experiment, just to cut a long story short, that they inhibit macrophage immunity against a tumour, and it switches them to something called it's a very broad term, but a term called the M2 macrophage. And that's just it just means a very broad polarization of the cell to do a certain type of thing. And in this case, it's inhibiting the immune response against the tumour. So it's allowing the tumour to grow much freer. And so have you come across as well, thinking about this at the same time, so there's parallels here with immunotherapies of cancer, current immunotherapies of cancer. So the classic two at the moment are antibodies that are directed against molecules called CTLA4 and PD-1. These are clinically used now, and they are practice changing in the sense that patients who respond well to these types of therapies have prolonged life expectancy, and they may in fact have, you know, progression-free survival. It may actually be curative. I mean, I think we still don't quite know that yet. So it's amazing, right? And so the Siglec and sialic acid interaction is actually a mirror of that. It's very similar.
Okay.
But it's got one slight difference, which is the CTLA4 and the PD-1 interaction is based on immune cells interacting with a tumor cell. So there has to be this interaction between the cells. With the sialic acids, these are not just membrane bound on a cell, but they are found within the environment, in the extracellular space. And again, you know, I sort of gave this analogy of this sort of forest, or this brush of glycans. So that's what it's like within a tumor. You've got this brush of sialic acids being expressed within a tumor, and you've got immune cells infiltrating. And when they interact with this brush of glycans via Siglecs, it's like a brake system. It's like the brakes on a car. So it just switches them off. So we kind of alluded to this at the end of the study, that if you inhibited this interaction, you could switch the immune system back on again. And so, you know, that was just some of our ideas from about 6 years ago or so. But since then, I mean, I haven't worked on this directly, but some of my colleagues have continued to work on that. But more excitingly, there's now 3 or 4 biotech companies which have taken this idea up, and they've got some really promising results in preclinical models, so not in patients yet, where they're actually targeting this interaction between Siglecs and sialic acids. And it's, you know, it's curative in an animal, but often it is. Okay, it's only when you move into humans that you have all the other complexities to deal with. So, but I mean, they've started now clinical. I think they're recruiting for clinical trials. So, I mean, good luck to them. I really hope that works out. That could have a big impact on cancer treatment. But we have to wait now 5 years or so to see.
Wonderful. I mean, that's absolutely fascinating and really exciting.
Yeah, it's great to have been involved in that. You know, quite a few years ago, and I'm really pleased to see that it's actually coming into some sort of fruition now, where it may in fact be changing how we treat tumours. I think we have to wait a few more years yet, of course, but I think there's high potential there.
So my next question is, what is the role of the sugar molecule found in red meat and dairy called Mu5GC as a contributing factor to carcinoma formation and growth?
Yeah, so this was also work that I was doing with Ajit Varki some years ago, and we were looking at this particular type of sialic acid called Mu5GC. So to give you a bit of background on this molecule, it's found in all mammals and some other animals as well, but not humans. And it turns out there was a mutation that occurred sometime in our ancestors, and it may perhaps have been in response to something like a COVID pandemic, which led to a change in the type of sugars that we have in our body, and so we lost the ability to make it. However, we have all of the machinery still present to process it. So if you think, then okay, if you have a diet where you eat other mammals, which most of us still do, and it'll be things particularly red meat like cows, pigs, some dairy products, maybe a little bit less in milk, then part of that diet is going to be this non-human glycan called Mu5GC, but our bodies recognize it as self. So we take it up and we express it on our cell surface. Now, it turns out that, that our immune system, on the other hand, doesn't recognize it as self, it recognizes it as foreign. So it generates an immune response against it. There's very good data showing that this happens about 6 months after birth, that you will start to have an immune response against new 5GC, and it coincides with starting to eat solid foods. So the estimate is that everyone wandering about today has somewhere between 0.1 to 1% of their total antibodies in their blood will be against the sugar molecule, and that's a lot. That's a lot actually to have constant. I mean, and obviously it depends. There's going to be people with none to low. I mean, if you've never had anything which has had new 5GC, and then you're not going to have that any immune response against it. So what's the problem with this? Well, if you're eating diets very high in red meat, where there is actually a link to an increase in risk in getting cancer, if we apply this idea of this new 5GC and antibody response again to this idea, what you've got is a situation where you've got a diet high in new 5GC. It's leading to tissues that are expressing new 5GC in your body, and in response your immune system is mounting a chronic response against it. So it's this constant immune response against the tissues, and this may involve things like reactive oxygen species, for example, which are things released by cells to normally to kill pathogens again. But in this case, it's kind of killing cells and it's damaging cells. And so over a long period of time of this happening, there's evidence to suggest that this might be leading to an increased risk of cancer. I mean, we did all of this in tumour models, not in humans, obviously, and it showed, I think, a 30% increase risk of getting cancer in the models that we were working with. We've not proven it in humans. And also, I would say, I mean, I still eat red meat. I mean, I still like a fry up and everything else. So it hasn't really stopped me eating meat, but it probably does say, that you don't want to eat stuff in excess.
Yeah.
Probably, but that's kind of a normal. That's something that is passed down anyway from generation to generation, that you should eat a balanced diet and the rest of it. And that's what the study says in the end, is that you should just eat healthily.
Yeah. I suppose it's really highlighted how much we can, we do have some control over our risk of getting cancer. So, smoking, and it's a choice. So
exactly. And the risks of red meat, I believe, are far lower than something like smoking. And not to downplay the importance of the mechanism that we found, but just to reassure anyone who's listening, thinking should they be eating meat. I mean, there are a lot of benefits to eating meat as well. So, but yeah, you're absolutely right. There are a lot of environmental factors that impact the risk of getting cancer. But it is just impacting the risk. There's still, of course, a chance that you will get a cancer, but you won't have done any of these things like smoking, etc., and you'll have lived a very healthy life. So it's more like rolling a dice.
Yeah. So my next question is, what is Versican as a component of the ECM, and how can it interact with inflammatory cells, activating signaling pathways that promote the synthesis and secretion of inflammatory cytokines? And how does it interact with growth factors as well?
So Versican is one particular extracellular matrix protein, and it's more thought of as being expressed in a developing embryo. So it's more of a developmental protein. And one of the things it does in development is help guide immune cells to go to the right place. So it protects tissues in some cases from immune cells. In an adult, Versican is present, so there is Versican expressed in all our tissues, but it's usually expressed in particular forms, not in particularly high amounts, and in very specific places. And it has quite a role in, again, it's mostly involved in, from most of the studies that we've looked at on it so far. And I must point out, actually, it's quite a new molecule for us to be studying. We don't have a big history with it, but there are labs that we work with who've got a very long history with this molecule. It seems to be involved mostly with regulating where immune cells go, right? So if you think about it, when you've got an immune response, immune cells have got to work out where to go. So they need roads or tracks or highways, whatever you want to call them, which they travel down to get through a tissue to the place where they need to get to. And I think Versacan is one of these proteins which does that. So it allows immune cells to traffic along and get to the place they need to get to. And one of the things she said was, how are cytokines and chemokines involved in this process? So that's getting into the molecular detail of how this molecule functions. So cytokines, chemokines, they're attractants in some cases. They stimulate and tell immune cells to move in this direction. So we often talk in biology about there being gradients of chemokines and cytokines. Now, how do you regulate these gradients? Well, that's where Versacan comes in again. So it has a particular types of structural motifs, shall we say, that are expressed on its surface, which are able to grab hold of growth factors and cytokines, chemokines, and they generate a reservoir which cells which are then coming into a tissue will sense and be attracted towards. So in terms of what's happening with them in a tumour, it could be, we currently think it's a little bit like a double-edged sword. So Versacan, as the name suggests, is a very versatile protein, and it has different functions. And some of those functions are actually sort of counterintuitive. So if I tell you one thing about Versacan, I can then tell you the other thing about it as well. So it can, in some cases, actually stimulate an immune response against a tumour, so it can be a good thing. But the type of stuff we're doing in the lab, our current thinking on it is that it's probably a bad thing. So what we think it does is it's forming these tracts within a tissue, telling immune cells where to go. We don't think it's telling them to go to the tumour. We think it's saying the immune cells to go everywhere but the tumour. So one of the questions we have in the lab is, well, what happens if you inhibit Versacan in some way, or you break it down? Does that then improve the immune cell infiltration into a tumour and lead to better tumour killing? That's a question that's ongoing, but that's the kind of idea that we have at the moment for Versacan.
Yeah. Absolutely. Well, that's all my questions covered, really. It's been wonderful and fascinating and really exciting. The opportunities for the future and future cancer treatments that may arise from some of this work that you're doing currently is amazing. So I guess to finish off, would you like to say anything about your most recent work, or if not?
No, yeah, I'm happy to. Yeah, no, I think I think to sort of sum up what we're trying to do in the lab at the moment, as I mentioned, it's all focused around the environment and particularly this extracellular matrix. And the way we sort of think about it is, sorry, I've just switched off my camera, it's back on. The way we sort of think about it is you've got to get good delivery of therapeutics into a tumour, and those therapeutics could be small molecule chemicals like classically used chemotherapies, all the way up to something like an engineered immune cell which is designed to go in and kill a tumour. You've got to get that delivery into the tumour. And in the lab, what we think of the extracellular matrix as is a final barrier. It's a final barrier through which all of these things have to move to get good killing of a tumour, so good therapeutic response. So we're hoping, our hope is that by doing this type of research, we'll not only understand more about tumours and how they build their environment, but we'll provide ways of breaking down this barrier. And it's through this barrier then that all of these wonderful therapeutics that have actually already been developed can hopefully start to really work as they were intended to. Time will tell, obviously, but that's the real hope of the lab.
Absolutely. I guess the efficiency of all the existing drugs and therapies that are out there could be greatly improved. So
Yeah, well, I mean, we may already have some really good therapeutics, and it's just about getting a better delivery of them into the tumour and knowing how to use them better. There's kind of a saying at the moment that the clinical cancer research work that's going on at the moment is moving so much faster actually than the bench side work, which is what I do. So one of the advantages of working in an institute like Bart's in London is that we work very closely with the clinician scientists as well. So that's very research enabling and helping us to move our work forward faster.
Amazing. Well, thank you so much for this today. I really, really appreciate your time. It's been wonderful.
It's a pleasure. Pleasure to speak to you, Martha.
Yeah, I've really enjoyed it. And for everyone listening, the papers that have been mentioned today will be linked next to, well, below this podcast now. So thank you so much for everyone for listening, and I will see you next time.